Hello and welcome to today's tutorial. I'm Carl Peters from BMG LABTECH, and I'll be giving you a little bit of information about one of our particular readers and its functions, and how that has helped the people at Promega to develop these walkaway cell health assays. So I'll keep my bit brief because I'm actually really interested to see what Andrew's been doing up in Wisconsin. So. So I'm going to be telling you a little bit about the Cleri Estar, and in particular, the capabilities that are enabled when this is matched with the Atmospheric Control Unit, or ACU. It's a multi-mode microplate reader, and all of these functions are capable within this reader. But I'm going to be focusing on fluorescence intensity and luminescence, as those are the detection modes that are going to be spoken about today. So the means by which the Cleri Estar can perform fluorescence intensity detection is through the use of a revolutionary type of monochromator. It's based on linear variable filters, and it allows for complete flexibility in adjustable band passes, anywhere from 9 to 100 nanometers. So in addition to selecting wavelengths, you can have that adjustable capability for your band pass. And we found that for the majority of fluorophores, having a wider and very specified band pass is very beneficial in terms of the sensitivity of detection that you can obtain. The light path in the Cleri Estar is an open-air optic system, and so it requires the use of dichroics. And for that purpose, we actually have an adjustable dichroic. So that means that the absolute best dichroic for your particular experiment to focus excitation light onto your sample and then allow the emission light to actually reach the detector, that can be adjusted and put in place regardless of what the fluorophore is. So with this new type of monochromator, compared to your traditional monochromator, we have much greater light transmission. So there's basically a higher transmission efficiency, and the amount of light that is able to be captured compared to what the excitation light is much better, and that is shown graphically here. And so what this means is better sensitivity in this type of reader. So what we have is, with this monochromator, a sensitivity of detection that's actually quite close to filter-based readers. And, but we do have the capability to use filters as well. So if you require the absolute highest in sensitivity, especially for cell-based assays or bottom reading, that this is a technology that you can employ as well. So it gives you the flexibility of the monochromator to maybe to perform some discovery, and then the ability to use filters if you want to have better sensitivity. The other detection capability that Drew will talk about today is luminescence. The Cleri Estar has high sensitivity luminescence with about 0.4 picomolar ATP LOD, and high dynamic range, so a wide range of signal that can be detected. So this is an improved dynamic range over our previous instrumentation, and is something that it makes it an instrument with very high utility. So as I mentioned, the capabilities can be enhanced with this atmospheric control unit, which allows you to control CO2 and oxygen. So by pumping in CO2 or nitrogen, you can regulate the gas levels within the reader between 0.1% up to 20%. This is the unit itself. It has a touch-based screen, so you can control it from there. But it is also integrated with the software, so all of the gas controls can be enabled within the software interface. And what this enables you to do is to use our scripting function, so that you can regulate the gas levels and change them over time if necessary, and to be able to study what effect that has. And all of this information is then going to be integrated into MARS, which is our data analysis software. And what that looks like is shown here graphically. In the upper panel, we have the actual O2 levels that were detected within the instrument, so the readout that's being enabled by the ACU. So you can see that the CO2 was kept constant, and it was taken through a series of steps to decrease the oxygen level down to about 0.5%. And this readout is a fluorescence readout that is a redox sensor. So as the oxygen levels go down, the signal intensity for this GFP probe is actually decreasing as well. And then as the concentrations of oxygen ramp up, that response is mimicked as well. So it's a very, I think, useful piece of equipment. And as Drew will go into, it enables you to truly walk away and perform continuous readings of your cell health assays.
Okay, so Carl told you about the Walkaway Automation part of it. I'm going to talk about, in very succinct examples, the real-time Walkaway cell health assay part of it. And I think that at least some of the screenshots will be exactly as they are represented in terms of the Mars software, as Carl talked about. So hidden beneath the elegance of these real-time plate-based cell health assays that provide quantitation as well as throughput is the fact that there's some basic cell biology happening. So in an untreated scenario, obviously you have healthy cells and you introduce a compound or a stimulus that may lead to either a non-proliferating cell type, whether in the case of transformed cell types that might be cell cycle arrest, which may ultimately lead to dead or dying cells by either apoptotic mechanisms or by alternative cell death mechanisms. What shouldn't be lost is the fact that this is both dose and time dependent, and it really depends upon the stimulus that you introduce. As I said, not only the strength, but the mode of action. And where this becomes difficult for employing endpoint-based assays are the fact that it's difficult to determine when to assess these changes in cell health. So we've worked very hard to introduce a suite of real-time assays which essentially take the guesswork out of this, where you collect data in a temporal fashion and you can measure these changes as they occur, with the understanding that each compound will have its own kinetic and cytotoxic profile. We've sought to characterize cell lines based essentially on three principal forms of assays. That is, viability assays that measure activities resident in viable cells, cytotoxicity assays which typically measure activities that are not found in viable cells, that are typically found as a result of loss of membrane integrity, found in the extracellular environment, and then also biomarkers associated with apoptosis, whether they be Cas space or annexin positivity or things like that. So essentially, as I showed you on the previous slide, the chances are is if they're cancer cells, they may be proliferating. In that case, a viability assay will show you an overall increase with time. If they're not proliferating, there might be either no increase or might even be a subtle decrease in activity. Or obviously in a dead or dying situation, that signal will decrease. Now this will all become important in terms of multiplexing and understanding the exact mechanism of action for the compound. As I said, cytotoxicity is an activity. These assays measure activities that are not found in viable cells. They're found activities that are found outside of viable cells. So for that reason, for proliferating cells or non-proliferating cells, there essentially be no change in signal until cells start dying when we see an increase in the signal. And that's the same with a mechanism of action cytotoxicity for apoptosis. So these Walkaway real-time assays are configured in the add, mix, and measure real-time format. So what does that mean from a workflow perspective? Essentially what you do is you seed the cell type of your choice into an assay plate. Of course, being cognizant of the fact of their replicative capacity. So if you intend to do a 72-hour study, you want to make sure that the cell health is maintained during that 72-hour time point with the culture conditions. If they're extremely proliferative cells, you might want to start on the low side, perhaps 500 or less cells. But if they're fairly slow doubling times 20,000 or cells. So that's something that can be empirically determined. After seeding those cells, if they're attachment dependent, you would obviously let them sort of acquiesce on the cell surface, attach, etc. Then you would dose the cells, you would add the real-time reagent, put it in the BMG with the ACU, and start collecting data. Before we move on to actual data, I wanted to show a typical plate layout. Essentially, what you would do is start on the left with the highest concentration of compound and do serial dilutions of that compound. Now, we found that 2 to 3-fold dilutions covers a lot of dilutional space, and for that reason is typically employed. Not least to be left out of this is that you absolutely have to have control wells which are untreated. And as I said before, if they have proliferative capacity, this would be something obviously you would want to understand in terms of a long-term exposure. What should also be included is some sort of a maximum treatment control, which is shown here in green. That's typically employed at the end of the 72-hour or 48-hour time point, but certainly could be a model compound that you know induces the phenotype that you're looking for, in this case, whether that be cytotoxicity or apoptosis. What we also like to include in this plate layout are no-cell controls, and these are essentially medium plus the reagents. Most of the reagents that we use to detect viability and cytotoxicity changes have some inherent level of either luminescence or fluorescence, which, based on the gain by the PMT, can be subtracted from the final results. So this is just a quick example of the script that we actually use. This is data from our plate reader. Using the ACU, one of the first things that you would do is make sure that you have the appropriate temperature for maintaining your cells, in this case 37 degrees. We have a 5% CO2, and that's essentially what our model system requires. Essentially what we do is we've created scripts that stitch together luminescence and fluorescent reads kinetically, and that's shown here by a script called Multiplex 48. We'll dig a little bit deeper into what that script looks like. Essentially, as I said, the first measure will be luminescence, the second measure will be fluorescence, and again, this is a format that we like, a multiplexed format, which allows us to parse the signals of a luminescent assay and a fluorescent assay as a function of time. We've set it up for 193 cycles over every 15 minutes, so for those that do math well, that's 48 hours plus an initial read. And we progress to other settings, so in this case we've selected the top optic for our luminescence reading, and obviously the gain setting is an important aspect of these particular measures. Lastly, because we've arranged it in multiples of the highest dose to the lowest dose, we've chose to read those signals so that our replicates are nice. Practically speaking, in 48-hour determinations, there shouldn't be that much of a difference between replicates when we're talking about a total read time of maybe 2 minutes for an entire plate. Nevertheless, for consistency's sake, that's the way we arrange our plates. Now, to talk about some of the real-time walkaway assays that we employ. We're very excited about the real-time glow MT viability assay, which measures the intracellular reductive capacity of viable cells. So this, for those of you who are not familiar with this assay, think of something like Alamar Blue or Cell Titer Blue or any of these MTT assays, these assays that measure reductive potential of viable cells. Our take is a little bit different in that we introduce a pro-luciferin substrate, which upon intracellular reduction allows a substrate for a unique and stable luciferase called nanoluc to be liberated to create light. Obviously, this is predicated on the fact that live cells are able to accomplish the reduction of the substrate, and the dead cells do not. One of the interesting aspects of this is that this reduction happens essentially in real time, and the molecules that are produced are consumed. So we essentially have an absolute real-time assay in that when cells die, so for instance, if you were to introduce something like a a lytic reagent, that light would go away because you no longer have the reducing capacity. So how does that look in terms of actual data? So again, these are screenshots from the Mars software collected at 4, 12, 24, 36, and 48 hours. Practically speaking, as I said, I collected it every 15 minutes, but that would be a kind of a sort of a busy slide. So I've segmented into convenient time frames. And essentially what you can see when you apply something like a histone deacetylase inhibitor, which is going to influence transcription and translation, we see that as a function of time, obviously we see here at 4 hours, there's not a lot happening, but as the untreated cells proliferate, that signal goes up because we have more cells as due to proliferation, whereas the ones that are actually toxic, if you take a look at the gray, that signal actually goes down because we've actually killed cells. So this is a very useful assay in terms of understanding not only the proliferative capacity or the cytotoxic capacity, but also these cytostatic effects, which are shown in the next slide. This is an example, again, a screenshot from Mars, which shows what happens when you apply staurosporin to K562s, which is an erythroid leukemia cell line, for a period of 48 hours. So each individual well has a trace of what's happening in terms of luminescence in those wells. At the highest dose, we don't see a lot of signal based on the scaling. We see a little bit of signal as we decrease that dose of staurosporin. And then ultimately, as I said, we always include the no dose control. So here's what happens at these highest concentrations. We see the signal come up, and once the compound essentially begins reducing the reductive potential of the probe, we see a decrease in that signal as a function of time. In this case, we can feel fairly confident that at 48 hours, those cells are mostly dead. So staurosporin at those doses has reduced the viability to 0 in all of those wells. In these mid wells, we essentially see, again, this ramp to steady state, and then there's essentially a long-term plateau, which is indicative of an anti-proliferative response, possibly cell cycle arrest, based on the known mechanism of action for this compound. At this dose, it is likely cell cycle arrest. In the untreated wells, we would expect a profile very much like this, where after we've reached our steady state and with each cell division over that 48-hour time point, we have essentially more. So taken together, these kinetic traces can be very meaningful in terms of the strength of the stimulus and the exposure of the stimulus. And obviously, you can always draw conventional EC50 type graphs based on that. Again, at no compound, we see a very steady increase. These are essentially, divisions of the cells over time. Another assay that we've been very successful with is an assay called Celtics Green cytotoxicity assay. It's also a real-time assay. This is an asymmetric cyanine dye which has an affinity for DNA. Our very talented chemists in San Luis Obispo have engineered it to be absolutely cell impermeable until a cell loses membrane integrity. So what this means functionally is that we don't have to worry about the dye leaking somehow into a healthy cell and essentially causing DNA adducts, which actually cause cytotoxicity. In all the cells that we've ever tested, you can apply this at the time of dosing, and over 72 hours, it doesn't get into viable cells. The way the assay works is we call it a profluorescent substrate in the sense that it has a very low fluorescent profile in the absence of binding to DNA, which is shown here. At 100% viability, we have low fluorescence, but there's a proportional increase in fluorescence as that percent of viability decreases. So that makes it very useful in plate-based formats. This is just an example. I think it also is shows the correlation between the abstract of a plate number and something visual This is an example of a microtissue spheroid, HCCT116, that had been not treated, a very compact spheroid. We do see specks of green, as you would in any particular growth situation where you will have dead cells, but obviously it's very pronounced when we introduce panobinostat, a histone deacetylase inhibitor, which essentially also destroyed the architecture of the spheroid, but also turned it green. So if you can imagine, what we're doing is essentially translating this greenness into fluorescence that we can measure in plate-based formats. Obviously, this would be a fairly low throughput type of an analysis, and a plate-based measure would be much faster. So how is this employed? We have two examples, one of staurosporine, this pleiotropic kinase inhibitor, and one using panobinostat, the histone deacetylase inhibitor. And the kinetics are actually very different, as well as the mechanism of action. So you can see, in the case of staurosporine, as we increase the concentration of staurosporine, we start seeing effects as soon as 8 hours, but certainly by 12 hours, and increasing thereafter. Now, I think it also should be said that if we were to employ endpoint assays in this sort of determination, if we were looking for something like lactate dehydrogenase release, this would require lots of effort and lots of time points, and probably people missing some sleep. This is something that we would just set up and collect the data as a function of time. I think it's also striking in the fact that at the highest concentration of staurosporine, at 50 micromolar, we see the start of toxicity beginning at about 8 hours, and it's completely maximized by 24 hours, as shown here is at the 48-hour time point, whereas at a lower concentration in the mid-range, 6.25, we're really starting to see the emergence of cytotoxicity away from the untreated values, which obviously there's a progression, a distinction at the 48-hour time point. So as I said, cytotoxicity is both time and dose dependent, and I think this is a very striking image of how those two interplay very well. In the case of panobinostat, this is an example, again, over 48 hours, where we've collected in essentially 4-hour increments. And you can see that we really start seeing, because of the mechanism of action of it being a transcription and translation poison, at about 20 hours is when we really start seeing this sort of cytotoxic effect, and over a range of different doses. So you can see, again, at very high concentrations or very low concentrations, the start of toxicity really begins at about 16 hours for all of these. Again, consistent with the mechanism of action for a histone deacetylase inhibitor. So these are just two examples of how dose and time dependency can be important for the interpretation of your results. Lastly, we're very excited about a new assay that is not commercially available, but which we expect to be available in the late spring or perhaps early summer. This is an assay called the Realtime Glow Annexin 5 apoptosis assay. So if any of you have done the annexin assay, essentially annexin is a protein that has affinity for phosphatidylserine, and it's been shown that during the apoptosis progression, phosphatidylserine will predictably flip to the outer membrane leaflet of an apoptotic cell in a predictable manner. And if the cell is not apoptotic, it will be retained on that inner membrane. So for this reason, in the case of typical annexin probes, you apply a fusion protein with annexin and perhaps some fluorophore. You do extensive washing, and you do either imaging or flow cytometry. Consistent with our dedication to developing real-time glow assays based around luciferase-type platforms, we've engineered annexin proteins to contain 2 parts of a binary luciferase reporter, annexin in the small bit and annexin in the large bit. And we can add these in solution to viable cells in the presence of a real-time substrate and a necrosis detection reagent. And if the cells are not apoptotic, these partners do not complement. It's only when we have phosphatidylserine on the surface, it drives the annexin fusion partners together, and thus complements the nanobit reporting system. And as I said, in the presence of our real-time substrate, it generates light in a glow-type fashion. Now, as I said earlier, apoptosis is distinct from true cytotoxicity in the sense that we still have an intact membrane. We just have PS on the surface. At some later point, due to secondary necrosis and time, we will see actual changes in membrane integrity, and we will get cells will stain the nucleus as well. And so this is somewhat analogous to flow cytometry in the sense that you always counterstain to understand the percent phosphatidylserine positive versus the number that are actually necrotic. Also, this is, as I said, absolutely homogeneous in that you introduce these reagents in solution at the time of dosing, put it in a plate reader, and measure. There are no washes or anything of that nature. So we can start looking at the induction of apoptosis in real time, which has been a very large challenge in terms of throughput. This is an example of applying bortezomib. Bortezomib is a proteasome inhibitor, and it essentially acts as the garbage disposal for misfolded proteins. When you perturb that activity, particularly with rapidly multiplying cells which have a tendency to produce misfolded proteins, you have cytotoxic events. So this is an example of applying the real-time and X-and-Glow assay chemistry to these cells that have been dosed, put it in a plate reader, collect at each concentration over 48-hour time point. Now in the gray, we see there really isn't much that happens for the first 8 to 10 hours, but by the 12-hour time point, we see an increase in this annexin positivity. So this means that we have at least a subset of cells that have phosphatidylserine on their surface. Our annexin proteins that have the nanobit technology have found that and are starting to create light. Now, at the same time, we have the necrosis detection reagent that tells us if we have dead cells, and we do not, essentially it's at the background. It's only at about 24 hours or perhaps 20 hours do we start seeing any appreciable membrane integrity. So this is obviously an event that precedes changes in membrane integrity, which again supports the notion that this is true apoptosis and not some sort of a alternative death mechanism or primary necrosis. But why I really like this graph is that it's striking in the sense that as apoptosis proceeds, we see not only an increase in the magnitude, meaning more cells that are involved, but we also see an increase in potency as a function of time. And you can see that even at 48 hours, we have a significant signal relative to some of those earlier induction events. If we were to do this assay with any of the wonderful caste-based activity assays that are available commercially, including our own, this would be a very difficult and time-intensive experiment to do, and frankly, I doubt that it would be doable in this sort of a format, certainly in a high throughput type manner. Another interesting part of this is that we can choose any particular concentration and take a look at the emergence of the PS positivity, in this example 1.1 micromolar in both the luminescence and fluorescence channels, and we can see that we really start seeing the induction of apoptosis somewhere in that 12-hour time point. It's more or less maximized by 24 hours and more or less sustained over that 48-hour time point, again, at that individual dose. And we see changes of membrane integrity beginning at about 24 hours. So again, these are real nice correlations for the appearance of PS and the actual cytotoxicity. Lastly, I'll end with a biologics application. This is an example of an experiment that we set up, and if any of you have done these sorts of experiments with chromium release or anything like that, they're absolutely toxic and not very fun assays to do. That's primarily due to the sensitivity. So this really speaks to the sensitivity of the assay, but also some of the things it reveals in terms of the kinetics. So this is an example of mixing natural killer cells that have been stimulated with IL-15 with the target cell population at different ratios. So at the highest concentration, we have 5 natural killers to 1 target as diluting. And we see very nice signals indicating that we've killed those target cell populations. In blue is the contribution of just the target cell, or excuse me, of just the natural killer cells, again, consistent with the way that we would conduct this experiment if we were doing something like chromium release. But I think the right-hand image is really the most striking, where we see the actual time when we're starting to kill. There is some natural killer acquisition. There are lots of receptor-dependent activities in addition to migration. And we see most of the killing beginning at about 30 minutes, and it's absolutely complete by 2 hours. So these were very effective killer cells, and you can see the contribution of luminescent signal from the target only, and the effector is fairly minor, again indicating that we have a true natural killer cytotoxic event. So, the conclusions that I've come to are that cell health responses to any stimulus are exposure and time dependent. I think we've done these lots of different ways over time, but I think that it's really time to start measuring these effects in real time, saving effort, resources, and money. And I stress effort, because any experiment that you can set up on a Friday and collect your data on the Monday morning is very useful in our laboratory, at least. Promega's real-time cell health assays and related multiplexes deliver dose-response information relating to proliferative, non-proliferative, and cytotoxic responses, and can help define mechanism of action when employed using the BMG Cleri Estar equipped with an atmospheric control unit. So these are pretty simple experiments to do, also in relation to the title about real-time walkaway assays. So once you have these set up, they're pretty simple to conduct. So with that, I'll open it to questions for Carl and I. Thank you.